Okay, in this video we're going to talk about nucleic acids. And nucleic acids are another class of biomolecules that are also composed of a lot of carbon, hydrogen, oxygen, nitrogen, and some phosphorus. And these are actually the largest molecules of the body. Now, these nucleic acid polymers are made up of these monomers called nucleotides. And these are actually made of a nitrogenous base, a sugar, and a phosphate group. And this is actually what makes up DNA and RNA. So DNA is deoxyribonucleic acid, RNA is ribonucleic acid. They're very similar in terms of their molecular structure, and they're involved with coding of genetic information in your body. Now, DNA holds this genetic blueprint for the synthesis of all of your body proteins. There are approximately about 10,000 different genes that you find within the DNA of your cells. And these are all organized with this double-stranded helical molecule we call the double helix that's located in your cell's nucleus. Now the nucleotides here contain this, remember, the ribose sugar, phosphate group, and one of four nitrogenous bases for DNA. We have adenine, guanine, these are examples of purines, and then cytosine and thymine, which are the pyrimidines. And it turns out that purines and pyrimidines can bond with each other to form this double helix. So adenine always binds with thymine, and guanine binds with cytosine, and they're actually bound through hydrogen bonds in the double-stranded helix itself. So, like I was saying, the bonding of nitrogenous bases helps to hold these opposite strands together, and the complementary base pairing is A with T, G with C. So adenine with thymine, guanine with cytosine, and they're held together by a series of hydrogen bonds. So if you look here, this is showing essentially the structure of DNA. And remember, DNA has your phosphate group here, the sugar group here, ribose sugar, and the nitrogenous base. So our nitrogenous base in this example is adenine, and this is one strand of the DNA molecule, or one side of the strand. And this is another side of the strand over here, which you can see our phosphate group, our ribose sugar, and then a different nitrogenous base, which is thymine. And you can see that the oxygen on this side is able to hydrogen bond with the hydrogen on the other side. And then the nitrogen group on this side is able to hydrogen bond with the hydrogen on this side. So you can see that these are held together with hydrogen bonds. If y'all remember, hydrogen bonds were actually a weaker type of bond. However, the many hydrogen bonds of a single DNA strand give DNA a lot of strength as a whole. Individually these bonds are pretty weak, but as a whole, DNA is held together very strongly. RNA is actually what links DNA to protein synthesis. So if DNA is like the blueprint, RNA is the transportable form of this blueprint that is able to carry that information to the site of protein synthesis. So RNA is this single-stranded linear molecule, and it's mostly active outside of the nucleus. It also contains a ribose sugar, but not deoxyribose, hence the name RNA. Instead of deoxyribonucleic acid, it's ribonucleic acid. And thymine is replaced with uracil. So we actually don't have thymine in RNA. Instead, we have another type of nitrogenous base called uracil. And there's three major varieties of RNA in our cells. We have messenger RNA, transfer RNA, and ribosomal RNA. Now we'll get into more detail on the functions of these in chapter 3 when we talk about the cell. Now, another class of nucleic acids is ATP. So ATP is actually the energy currency of your cells, and it's actually how chemical energy is captured in this form, adenosine triphosphate, or ATP. So ATP is actually used to directly power different chemical reactions in your cells. It offers an immediate usable form of energy for your body cells. And the structure of ATP is you have adenine with RNA nucleotide and two different phosphate groups. Now what happens is that between each of these phosphate bonds, they're high energy bonds that can be used for different types of work within your body cells. So this is just showing the general structure of ATP. So remember, you have the adenine bound to a ribose sugar and then three phosphate groups. Now these high energy phosphate bonds are the ones that can be hydrolyzed to release chemical energy that can be used for a variety of different body processes. Now when you cleave one of these phosphate groups and this phosphate is left, what you end up with then is ADP, which is adenosine diphosphate, And if you cleave the second bond here, and you'll have one phosphate group left, then it's adenosine monophosphate. Otherwise, if there's no phosphates on this at all, and you just have adenine and ribose, we just call these adenosine. But you're essentially just seeing what gives the molecule its different names, depending on how it's structured. Now, ATP, the terminal phosphate group can be used, transferred to other compounds that can be used to, store different energy, or you can use that chemical energy for work. Now, the loss of one phosphate group converts ATP into ADP, remember adenosine triphosphate, into adenosine diphosphate, and the loss of a second phosphate group converts to ADP to AMP, or adenosine monophosphate. Okay, so this slide is showing the interconversion between ATP and ADP. And you can see that ATP, or adenosine triphosphate, can be cleaved into adenosine diphosphate. And it turns out this is a hydrolysis type of reaction where ATP can be hydrolyzed into ADP, and that chemical energy that's released by cleaving a phosphate group away from ADP actually releases some energy as well. Now, a majority of this energy is lost in the form of heat, which is basically infrared radiation. However, some of that energy that is released when the chemical energy released when these bonds are broken can be used for work. Something close to about half of that energy can be actually used for work. Now, the reverse is possible as well, where if you put a little bit of energy into this chemical reaction, and this could occur in mitochondria, where you can use chemical energy from other molecules to make additional ATP through dehydration form of synthesis. Now, ATP can be used in a variety of different ways. So we can see it's used for transport work, where you may want to transport molecules inside your cell out of the cell by actually physically moving them through a transport protein in the plasma membrane. ATP can also be used for mechanical work, where the chemical energy can be used to effectively power the contraction force of cells, and that can generate tension, which could be used for different types of mechanical work, like moving digestive contents around, or regulating blood pressure, or even moving your bones around. Now, ATP can also be used for chemical work, where you might want to have anabolic reactions where you can take monomers of something and make a larger molecule. Now, some of these reactions actually require additional energy input, so that ATP can be used to make biomolecules in the body.